Welcome to this lecture on follicular development. Imagine a massive greenhouse filled with thousands of tiny, dormant seeds. Every month, a small batch wakes up, but only one champion grows large enough to produce a beautiful flower. In the human body, the ovaries are exactly like this greenhouse. And today, we are going to explore the remarkable biological and mathematical journey of how a single microscopic follicle matures to release an egg. To understand follicular development, I want you to imagine a massive greenhouse filled with thousands of tiny, dormant seeds planted in the soil. Every month, the gardener turns on the water and nutrient supply, waking up a small batch of these seeds. As they start growing into small plants, they compete fiercely for sunlight and resources. Ultimately, only one champion plant grows large enough to produce a beautiful, fully mature flower. In the human body, the ovaries are exactly like this greenhouse, and the seeds are tiny structures called follicles. This monthly race is what we call follicular development. Now that we have our greenhouse analogy in mind, let us look at the actual biology. What exactly is a follicle? In human physiology, a follicle is essentially a microscopic sac located deep within the ovary. Inside this sac lies a single immature egg, which we scientifically call an oocyte. But the egg does not survive alone. It is surrounded by specialized supportive somatic cells, primarily the granulosa and theca cells. You can think of these surrounding cells as the egg's personal life support system, providing the nutrients, environment, and signals it needs to survive and eventually mature. Understanding what a follicle is brings us to how it grows. This maturation process, formerly known as folliculogenesis, does not happen overnight. It progresses through several distinct anatomical stages. Every follicle starts its life as a tiny, dormant structure called a primordial follicle. As it wakes up and begins to develop, it transitions into a primary follicle and then expands further into a secondary follicle. Finally, after intense competition for resources, usually only one champion reaches the fully mature stage, known as the tertiary or Graafian follicle, which is fully prepared to release the egg. So, what drives this incredible growth from a primordial to a Graafian follicle? The entire journey is tightly controlled by chemical signals from the brain, specifically follicle-stimulating hormone, or FSH, and luteinizing hormone. As FSH stimulates the follicle, the supportive granulosa cells multiply rapidly. As this cell population explodes in number, they begin secreting massive amounts of estrogen into the bloodstream. This rising estrogen is crucial as it prepares the rest of the female reproductive system for a potential pregnancy, signaling that a mature egg is almost ready. Before we look at the mathematics of this growth, we must understand why it matters clinically. Follicular development is absolutely critical because it is the foundational engine of female reproduction. Furthermore, these growing follicles act as temporary endocrine glands. The estrogen and progesterone they produce regulate the menstrual cycle, maintain bone density, and support heart health. When this precise biological process malfunctions, it can lead to severe conditions like polycystic ovary syndrome or infertility. Therefore, mastering this concept allows doctors to design contraceptive methods, manage hormonal disorders, and treat infertility effectively. This rapid multiplication of granulosa cells we just discussed is not random. It follows a precise mathematical pattern. Because each cell divides into two, the population experiences classic exponential growth. We can model this using the formula n of t equals n naught times e to the power of r times t. Here, n of t is the total number of cells at a given time. N naught is our starting population. The constant e is the base of the natural logarithm, which perfectly describes continuous biological growth. The variable r is the growth rate, which increases when hormone levels are high, and t is the time elapsed in days. If hormones drop, r becomes negative and the follicle shrinks and dies. Before we move on, let us see this formula in action with a concrete example. Imagine a secondary follicle begins its rapid growth phase with an initial population of 1,000 granulosa cells. It has a continuous growth rate of 0.2 per day. We want to find the total cell count after 10 days. We start by substituting our known values into the exponential equation. The exponent becomes 0.2 times 10, which simplifies to exactly 2. We then evaluate e squared, which is a known mathematical constant approximately equal to 7.389. Multiplying this by our initial 1,000 cells gives us a final answer of 7,389 cells. Physically, this massive seven-fold increase explains why the follicle swells so dramatically in size. Now that we have successfully used this formula to find the final population, it leads us to an important question. What if we already know the target population required to trigger ovulation, and we need to find out how long it will take to get there? To do this, we must mathematically isolate the time variable, t. We start with our standard exponential growth equation. Because t is trapped up in the exponent, our first logical step is to move the initial population, n naught, to the other side. We do this by dividing both sides by n naught. This gives us a ratio of the final population to the initial population on the left equal to the exponential term on the right. Continuing from our previous step, we still have the time variable trapped in the exponent. To bring it down to the main line of the equation, we apply a specific mathematical operation, the natural logarithm, to both sides. Because the natural logarithm and the constant e are inverse functions, taking the log of e effectively cancels out the base. This beautifully simplifies the right side to just r times t. Finally, we divide by the growth rate r. We have now proven that the time required to reach any critical mass is equal to 1 over the growth rate, multiplied by the natural log of the population ratio. This derived formula is incredibly powerful for predicting ovulation. Let us apply the logarithmic derivation we just proved to a real physiological scenario. For a dominant follicle to trigger ovulation, it must reach a critical mass of 50,000 cells. It currently has 5,000 cells and is growing at an accelerated rate of 0.46 per day. We need to find the time, t. We substitute our values into the equation. Dividing both sides by 5,000 shows the population needs a 10-fold increase. Taking the natural logarithm of both sides brings our exponent down. The natural log of 10 is approximately 2.30. Dividing this by 0.46 gives us exactly 5 days. Physically, this rapid 5-day growth spurt ensures the egg is perfectly mature and ready for release. Now that we have calculated the massive cellular growth required, let us look at what this actually looks like inside the ovary. On screen is a schematic showing the anatomical progression of a follicle. On the far left, we see the tiny primordial follicle, dormant and waiting. As follicle-stimulating hormone, indicated by the arrows, begins its work, the follicle transitions into the primary and then the secondary stage. Notice how the layers of supportive granulosa cells multiply, exactly as our exponential math predicted. Finally, on the right, we see the massive Graafian follicle. It has developed a large, fluid-filled cavity called the antrum, pushing the mature egg to the side, perfectly primed for ovulation. We have covered the full journey of the ovarian follicle today. Let us review the most important ideas. First, follicular development is the highly coordinated maturation process that transforms a dormant seed into a mature, egg-releasing structure. Second, this journey is driven by the rapid, exponential multiplication of granulosa cells, which we can accurately model and predict using mathematics. Third, this entire process is tightly regulated by hormones, like FSH, ensuring the survival of the fittest follicle each month. Understanding this beautiful intersection of biology and mathematical precision is the key to grasping how the female reproductive system sustains fertility and overall endocrine health. Excellent work today.